Good morning. In this ninth video on the cell, we are going to discuss in detail the types of transport through or across the cell membrane. In this diagram to the right, you can see two types of passive transport. You can see simple diffusion and facilitated diffusion. And whether it is simple diffusion or facilitated diffusion, since it lies under passive transport, then it does not require energy, and it is the transport from a higher concentration where the particles are present in a higher concentration to where the particles are present in lower concentration. If that process of passive transport that occurs down concentration gradient occurs without the need of a channel nor the need of a carrier, then it is just simple diffusion. And in those channel-mediated passive transports or carrier-mediated passive transports, only one type of particles is transported, meaning that they are specific to a particular particle. For example, if this channel is a sodium ion channel, then it will be specific only to sodium. And if this carrier is a glucose carrier or a glucose transporter, then it will be only specific to glucose, meaning that it will allow only the passage of glucose, and that's why we call it a uniport. In active transport, we have several types, or actually we have two broad categories of active transport, but let's agree first that all types of active transport that, as you can see on the diagram, involve passage of the particles of the substance from where they are present in lower concentration to where they are present in higher concentration. And therefore, even here, when low is at the bottom, it is again from low to high. So what's important is the direction of the arrow. It's not important whether the higher concentration is extracellular or intracellular, but what matters in active transport is to find out that the passage of particles is from lower concentration to higher concentration. So this is a common feature to all types of active transport. In primary active transport, we have two types of transport, of primary active transport. And when we say primary, we mean it occurs on its own without being dependent on a previous kind of transport. So it is primarily a means of transport that does not depend on the occurrence of a previous transport. Primary transport can be uniport or unitransport and co-transport. As its name indicates, co-transport means that you transport two substances simultaneously at the same time. For example, in uniport against concentration gradient, for example, when a pump, for example, in such a case, of course, the protein that allows this type of transport is called a pump. So it is a pump-mediated transport. So all types of primary active transport, whether they are uniports or co-transports, they are pumps. When we use the verb pumps, we mean to transport against concentration gradient. But not only this, not only against concentration gradient, but using chemical energy. And that chemical energy is derived from the hydrolysis of ATP into ADP, or adenosine diphosphate and inorganic phosphate. The pumps that carry out co-transport will transport two substances at the same time. But in both cases, we will have transport from lower concentration to higher concentration, meaning that both ways or both types of ions, for example, are carried against concentration gradient. So if it is this pump, if it is what we call the sodium-potassium pump, so it will transfer potassium inwards against concentration gradient and will transfer sodium outwards against concentration gradient. So here, for example, we are going inwards, but from low to high. So this is potassium that is carried inwards because potassium should be the main cation intracellularly. As we know previously, in order for the cell to function properly. So why does potassium have to be the main cation inside the cell, or intra means inside, intracellularly? This is for proper functioning of the cell. Meanwhile, sodium is pumped outside the cell, again in an active transport process from low to high. Again, why? Because sodium has to be the main cation outside the cell, or the main cation extracellularly. And why does it have to be the main cation extracellularly? For proper functioning of the cells. So this is a kind of co-transport because we are transporting both sodium and potassium at the same time. It is a type of active transport because we are transporting against concentration gradient using energy. And then we have the secondary transport. And secondary transports are mediated via carriers, not pumps. We call the protein that mediates them a carrier. So here we are passing from low to high concentration, meaning that this process is an active process. But on the other side, we have passage of substances from high to low. So this is a passive process. So unlike the sodium-potassium pump, which is this one we've just studied, we call it a sodium-potassium pump, and it is present in all cell membranes. And by the way, 2 potassium ions are pumped in for 3 sodium ions out. And this difference in the number of cations that are pumped in and out is meant, and it is what creates a potential difference across the cell membrane. Because when you pump 3 sodium ions outwards and simultaneously you pump only 2 potassium inwards, you are ensuring 2 things. First, that you are ensuring that potassium is the main cation intracellularly, which is good for the cell, and that sodium is the main cation extracellularly, and this is again good for the cell. But you're also ensuring that the positivity outside is always higher than the positivity inside, or in other words, that the outside of the membrane is always positive in relation to the inside. When we draw this, we don't mean that the overall inside is negative, but we mean that the overall inside is less positive, at least less positive than the outside. So there is a potential difference that's being created, and that potential difference is what we call the membrane potential, and it is actually a resting membrane potential, because if the cell is a nerve cell and it is stimulated by a nerve impulse, that difference in potential will be disturbed. So this is what we call the resting membrane potential. We will see examples of antiports. This is called an antiport because the ions are in opposite directions. The co-transport here again is an antiport. So this one can be considered a co-transport, but also an antiport. And we will see how a substance is pumped in inside the cell actively in order for another substance to pass passively from the inside to the outside. We will see details of this later on. In symports, we have the 2 ions are, or the 2 particles are transported in the same direction, and one of them is transported from low to high, which is an active transport mechanism, and the other one is transported from high to low, which is a passive transport mechanism. This is again a co-transport. Both carrier-mediated ones are co-transports because they occur at the same time. But in antiport, the two particles are transported simultaneously, whereas in symports, both are types of co-transport, the antiport and the symport, meaning that they are transported at the same time. But in antiport, the two particles are transported in opposite directions, whereas in symport, the two particles are transported in the same direction. On this slide, we will be discussing the diffusion of substances through channels, or the diffusion of ions specifically through channels, which is called the facilitated diffusion. So facilitated diffusion is a type of passive diffusion, meaning that although it requires a channel, yet it requires no energy. And it requires no energy because the ions are being transported from higher concentration to lower concentration. But why do we say that it is facilitated? Facilitated means that it is being made easier. And it is mainly being made easier through the hydrophilic protein channels, because as you know, those charged ions like sodium and calcium cannot go easily through the hydrophobic interior of the cell membrane or the hydrophobic lipid tail. So we need a protein channel that is more or less like this, and it is hydrophilic from inside and hydrophobic from outside. So it has hydrophilic domains on the inside of the channel that match with the hydrophilic ions, with the ions and the charged particles. But on the other hand, the outer part is hydrophobic in order to match with the surrounding hydrophobic interior. And it is used to transport single ions, and that's why we say that it is specific. So every channel is concerned with the transport of only one type of ions, but not the others. We have two types of ion channels. We have open ion channels, as we said before, that are constantly open, and we have gated ion channels. And the gated ion channels are not constantly open. This is what characterizes the gated ion channels. They are not constantly open, but they open and close according to certain criteria. For example, according to the electric gradient. And when we mean the electric gradient, we mean the potential difference across the cell membrane, or it depends upon the chemical gradient, meaning how is the difference in concentration between the outside and inside. Is it big enough or not big enough? And at a certain chemical gradient means difference, or at a certain potential or electrical gradient, there will be the decision to allow to open the channel and allow the passage of ion. So for example, here we can see that in this voltage gated ion, we have the membrane potential, the transmembrane potential, or the potential difference across the membrane is minus 70 millivolt. This electric gradient or potential difference does not allow the channel to be open. So we have like a small protein molecule that closes the channel. But when the voltage becomes minus 80 millivolt, for example, after electric stimulation, when a nerve cell undergoes electric stimulation, we will see that the voltage is instead of being minus 70 millivolt, it starts to diminish and become minus 60 millivolts, diminish in its absolute value. And in such a case, this closure by the protein is removed, and we can see that the ion can pass. So this is an example of what we call voltage-gated channels. The general term is gated, but when it depends on the voltage difference or the potential difference, we call it voltage-gated. We have another type of facilitated diffusion, which is the facilitated diffusion of molecules through the carriers. And the carriers, as we said before, are designed to allow the passage or diffusion of, and we say diffusion because it is a passive process, but in such a case, we have larger particles, which are usually molecules. Not only are they large, but also polar. For example, sugars, and of course, mainly glucose, which is the most abundant carbohydrate in the human body, and amino acids. So sugars and amino acids are characterized by being large molecules and polar molecules, and they require a carrier. And a carrier is nothing but a membrane protein. So if you're ever asked to define a carrier is a cell membrane protein. And that cell membrane protein is used to allow or to facilitate the transport of large polar molecules like amino acids and sugars. And it does this by changing its shape, by changing its shape. So it is an allosteric molecule. So here you can see the yellow molecule, which is the large polar molecule that we want to transport. So it binds with a specific site on the carrier. The carrier is open on the extracellular fluid side, and then after having bonded with a carrier molecule, the carrier closes from the extracellular side and opens from the intracellular side, and the large polar molecule is delivered. Another more complicated type of passive transport is the countercurrent exchange. And the countercurrent exchange is a special type of simple diffusion. And we have talked about countercurrent mechanisms before in the kidney when we studied the structure of the nephron, which is the functional unit of the kidney. And we saw that inside the renal tubule, we have plenty of such U-shaped loops that are called the loops of Henle, based on the scientist or named after the scientist who described them. And in the loops of Henle, we find that the fluid that has been filtered moves first downwards and then upwards in those two arms. So you usually have two arms, and you have fluids that are moving in opposing directions, and they should be very close to each other. This is the basis of any countercurrent mechanism, whether it is a countercurrent mechanism in the human body, like the loop of Henle in the renal tissue or in the kidney, or whether it is a countercurrent system outside the human body in an industrial field, for example. These are the properties, the main characteristics of a countercurrent exchange. But what is the benefit of a countercurrent exchange? It is to maximize the function. For example, the function of the loop of Henle in the kidney, the one we have just described, was to concentrate urine. To concentrate urine. How? By returning water to the blood. by reabsorbing or returning water to the blood. So transferring water from urine to blood, we are thus keeping our water, preserving our water, and meanwhile concentrating urine. So whenever you have a countercurrent exchange or a countercurrent system, the main importance is to maximize the original function for which the countercurrent system has been designed. The countercurrent exchange is a special type of diffusion, and since it is a special type of diffusion, then it is a passive process, and it does this amazing function of maximization of the original function without the expenditure of energy. And it involves the flow of adjacent fluids in opposite directions in order to maximize in the human body, to maximize the rate of simple diffusion of the substance. The example we are going to explain is what the countercurrent system that is present in the fish gills. And in the fish gills, if you remove of course, you know that if this is the body of the fish, so here we have the head, and on the other extremity we have the tail, here we have the eyes. And then if you remove that cover, which is the gill cover, you will see that you have like slits that consist of blood vessels, and though they are shelf-like and they consist of blood vessels, and these blood vessels are covered by mucous membranes, and in the blood vessels you will have blood, of course, that carries oxygen to the tissues. So what happens is that the fish swallows water, and that water contains oxygen. So water flows over the gills. If we change the color of the arrow in order to let's represent water by a different color than red, because so we will represent water by a green arrow. So water will pass in that direction towards the tissues in between the gills in special tissues. But meanwhile, we will find that the blood will pass the direction of flow of blood as the cardiovascular system or the heart pumps blood towards the head of the fish. So actually you have blood that flows towards the head. So we have like this, we have a fluid, which is blood, that flows towards the head, and another fluid, which is water, which flows towards the tail. And they are in close proximity to each other. So here we have the water which flows away from the head and towards the tail. So here we have all the criteria necessary for a countercurrent exchange to occur. We have two fluids that are flowing very close to each other, parallel to each other in two opposite directions. We forgot here to say that amongst the criteria of a countercurrent exchange is that they should not be divergent or convergent. They should be parallel to each other. So of course you can see on this diagram how water enters from one side, and this water, you can see how this water is high in oxygen, and of course this comes from the direction of the head. So this water that is colored blue comes from the side of the head and is full of oxygen, whereas the blood is coming towards the head. And this blood that is coming towards the head, actually, we had better draw it in blue, because it is blood that has a low oxygen content. It has a low oxygen content, so it is deoxygenated. oxygenated blood coming from the tail. What's the importance of specifying this? The importance is that this blood that is coming from the tail to the head carrying all the carbon dioxide is deoxygenated, meaning that it is poor in oxygen, as you know, whereas the water that is entering just comes from water and oxygen is rich in that water. So we are expecting oxygen to diffuse from the water to the sites where the blood is flowing through the head in order to enrich with blood with oxygen. So here we have seen by structure how this is a countercurrent exchange. But on the next slide, we would see how it maximizes the function. And the function here is to enrich the blood with oxygen. So in a countercurrent exchange, blood always comes in contact with water that always has a higher level of dissolved oxygen. So this structure in the fish gill allows this. So this countercurrent exchange structure allows this function. What is the function? It is that blood that always comes in contact with water that has a higher level of dissolved oxygen, and therefore there will always be a diffusion gradient for oxygen. And the importance of that structure is that this diffusion gradient is maintained along the whole length of the gill structure. This is the most important statement, because you might have a concentration gradient, but that concentration gradient might fade away or disappear very rapidly. So this is against the countercurrent system. But the countercurrent system allows this gradient or this difference in oxygen, this oxygen gradient, to be preserved along the length of the gill. So if you preserve it, then you ensure continuous diffusion from water to blood. You ensure continuous diffusion from blood to water, and therefore you are maximizing the function. But if, from water to blood, from water to blood. But if it fades away rapidly, then this is not maximization of function. So diffusion of oxygen will continue from water to blood as long as this gradient does not level out, and this, of course, maximizes gas exchange across the gill layers or the gill lamellae. But on the other hand, if you had a parallel flow instead, the other type of flow where you don't have a countercurrent exchange is called a parallel flow. And this is a bit confusing because we've just said that in a countercurrent exchange, the two vessels are parallel to each other. But by saying parallel flow, we mean flow in vessels that are parallel, but that where fluid does not flow in opposite directions. So they're parallel, that's true, but the fluids in both arms flow in the same direction. So the fluids in the two arms are in the same direction or flow in the same direction. So what happens is that leveling out of the concentration gradient occurs very rapidly. Your blood or the fish blood will be saturated with oxygen rapidly. Equalization of concentration between blood and water will occur rapidly, and the process will stop once equalization occurs. So here, when it is the same current, we start by water having 100% and blood having 0%. But notice that although the two vessels are parallel to each other, yet they are flowing in the fluids are flowing in the same direction. So gradually, oxygen is passing down concentration gradient until equalization of oxygen occurs, and when equilibrium is reached, diffusion is stopped because there is leveling out of the level of oxygen. But in countercurrent flow, where water enters the gills, saturated with oxygen, with 100% oxygen. And of course, this oxygen level declines gradually because of the diffusion of oxygen from the water to the blood, but it never reaches zero. Why? Because at the beginning, we have a high concentration of oxygen here and a low concentration of oxygen in the blood. So oxygen diffuses at a rapid rate. But since since blood is flowing in the opposite direction, even after leveling out here at 10, which is what is expected from the two parallel vessels, it will return back. It will backflow. And in such a case, 10 will be lower than 30. So oxygen will diffuse again to make this 10 20. 20 is lower than 50. So oxygen will diffuse again to make this 40 instead of 20. So this continuous closed loop allows the continuous exchange and the maximization of diffusion. The last type of passive transport we will talk about is osmosis. And osmosis is actually, as we said before, a special type of diffusion because it is diffusion of water. But diffusion of water from where to where? Diffusion of water from higher water concentration to lower water concentration. So it is essential here when you define osmosis to talk about water concentration, because if you just say concentration, then you are meaning the concentration of a solute. Say, for example, that we are having this solution that I'll be calling solution A that has, for example, 8 molecules of water and 1 particle of glucose, or 1 gram of glucose dissolved in 1 liter of water. So the concentration is 1 gram per liter. This is solution A. But if we look at solution B will have 3 grams of sugar per liter. Of course, since it has more of the solid particles, it has more of the solute, then it will have fewer molecules of water per unit volume. This concentration B, if we are talking solute-wise, this concentration B or this solution B is more concentrated than solution A, because if we keep the volume constant, for example, 1 liter, we will have more solute particles per unit volume. So it is a more concentrated solution, but it might have a lower number of water molecules, and therefore solute concentration differs from water concentration. When you say concentration, you're talking about solute concentration. But if you want to talk about the concentration of water molecules, then you say water concentration. Of course, you know the definitions. A solvent is a substance that helps dissolution. A solute is a substance that dissolves in the solvent. When we say hypertonic, we mean hypertonic in relation to another solution. So it is a solution that has a higher concentration of solid particles or solutes than another solution. So solution B is hypertonic to solution A. A hypotonic solution, again, hypo means lower. So it is lower than another solution. So it has a lower concentration of solute. So solution A in that diagram is hypotonic or has a lower solute concentration than solution B. If both solutions have the same concentration, like the same mass of solute per unit volume, then in such a case, we say that the two solutions are isotonic.